go back over it on Thursday. But also, I don't know why nobody's going to Dr. Welsh's study hours. Last semester he had 10 or so going all the time. So just because he's recording and posting it, that doesn't mean it's worth his time. Okay. So
Oh yeah, I'll put it up.
So I'm not sure why, if, you know, he changed it from Friday to Sunday and then nobody came on Sunday. So I am going to tell him to stop doing those unless people show up, because it's not really worth his time. You know, it's hard for him to do it, and he's doing us like a huge favor, and he does get paid, but very minimally, and he's like the craziest, busiest person. He does it because he loves to do it. So if you guys are gonna show up on Friday, tell him you wanna do Friday. If you're gonna show up on Sunday, tell him you wanna do Sunday. If not, all of you can show up on Sunday. Everybody text each other or send each other a message and say, you guys go this week, we'll go the next week. Something like that, to where he's actually engaging with students. Because if you're gonna just listen to him lecture again, you can just listen to me lecture again. You know, it doesn't seem like it's worth his time. So make sure that somebody shows up. If they don't show up, he's gonna log on, nobody's there, sweet, he's off, and there's no recording. So, and there's no extra credit, anything like that. So somebody's gotta show up to that Sunday, or else I'm just gonna tell him not to do it, because he's actually doing us a huge favor, and I don't think the recording, I mean, you might as well just watch a YouTube video, you know? So that is for questions and answers. He's got great information. He's a really great microbiologist. So if you guys have questions, like, that's a great time to ask them, instead of, you know, I'm happy to answer any questions at 12:30, and sometimes questions will just come up again before we meet. But that's a great time to ask him the questions, and then he'll answer those questions. Did you have a question?
Yeah. So I've been wanting to go to this, but I work like 12-hour shifts on the weekends. So it's kind of a damper. So what time is he doing it on Sundays now? Because I didn't even know they changed it.
Oh yeah, he changed it from Friday to Sunday because no one was going Fridays. Everybody said they couldn't do Fridays, so he was like, sweet, this week I'll do Friday and Sunday. No one came to Friday and no one came to Sunday. He was like, what in the world? So I was like, I'm sorry, like, I just felt really bad, and he's so busy.
That was my first time, honestly, that was my first time. I didn't, you know, I needed.
Okay, okay.
He met with me for like 2 hours.
Oh, awesome.
My kids were screaming in the background, and like, you know, people were not, I'm like, leave me alone. It was really cool.
Oh, good. Okay, yeah,
Sorry.
you know, he is golden. So if you guys
Good.
can text each other, just at least someone or two people kind of tag in and make the recording, and that will help other people. Then if you're like, well, I can't do next Sunday, get a couple other students to do it for you, just so that he's not wasting his time. Okay, so that's it for Dr. Walsh. So, okay, so we're gonna move through control today, and we'll try and power through, and then if you guys have any questions, please write them as we go, and let's just do a whole question and answer. Yeah?
One last clarification
Oh,
on
great.
that. This week is the plan to do one or the other, do you know?
I'm
Or
sorry, this week the plan is just Sunday.
Just Sunday, okay.
Yes, yeah, Sunday, 6 o'clock, right? Sunday, 6 o'clock.
PM?
PM.
Oh,
And,
okay.
yeah, and then just if there's a well, you know, if you can't go to the Sunday 6 PM, have some of the other students go so that they can record it. Or if there's a better time for you and a couple other students together, he can do a different time too. So, yeah, throw it out there. Okay, awesome. Yeah, I just want to be respectful of his time too, because he's like, you know, he's like in organic chemistry and calculus and linear algebra right now. I don't even know what he's in, but yeah, he's crazy busy, but he is a great tutor, and he loves to do it. So, okay, so let's talk about control. So we have talked a lot about bacteria. We kind of introduced control a little bit, and we talked a little bit about why we talk about control. We talk about control to take care of our patients. We talk about control because the hospital cares deeply about controlling pathogens, and infectious disease. They have actually, if you're ever interested in working for hospital administration, they have a whole infection control department. So their whole infection control department is usually staffed by a microbiologist or some, you know, an RN or something that is interested in microbiology or has done some work in it. And they are great. It's a great job. It's, you know, I'm guessing they make $150,000 a year is probably what I would, a wild guess. And they, so there's a whole team and a whole department at the hospital that does that for a living to make sure that there isn't infectious disease that spreads around the hospital. You know, every once in a while you'll get a patient, especially in Sacramento, that comes in with TB or comes in with some weird thing that somebody doesn't know what it is. Imagine how crazy the infectious control department was during COVID when we didn't know how it was spread. We didn't know about the HVAC. We didn't know if it was the cleaning crew. We didn't know, you know, if we were exposing our entire staff at the hospital, that kind of thing. So, they have an important job. There are some terms. So there are just some basic terms, and some of the students kind of get wrapped up in what I would do if I were to memorize these terms is just give myself 1 to 2 words of what they mean. And so then you're just clear of just taking 1 to 2 words out of what seems important to you and explaining what they mean. So one thing that I wanted to mention is the order of the terms. So decontamination, disinfection, and sterilization, it goes in that order of stringency. So this is less stringent. Will be less likely to kill organisms. This is the most likely to kill organisms. But why can't we just always use this? Why can't we, in a hospital setting, always use the most stringent that's going to kill the most organisms?
You can't use it on everything.
Yes.
You can't autoclave a bedroom.
Yes, you can't autoclave a bedroom, you can't autoclave a person, you can't autoclave their skin, you can't autoclave, there are plastic materials that will compromise the plastic if you make them too hot. There's all kinds of, this is the deepest, craziest topic you will ever hear about when you dive into that in the hospital setting. What they clean the bed sheets with, what they clean the mattresses with, what they clean the floor with, what they, the pens and the masks and the gloves and the, just the craziest amount of information about what can be handled with what kind of material. Also, we've been finding out that the more stringent that we go with the materials that we use are harmful to the patient's lungs or harmful to the healthcare workers or the cleaning crew who uses that all the time and now has lung damage from using these cleaning products. We had that issue in our lab as well, that we were using some sort of cleaning product before the one that we use now, and it was causing respiratory distress in some of the students or lab techs. So we had to switch to a less stringent type of cleaning fluid. So, it's just a lot to think about, but this is the order that it goes in. It starts with decontamination, goes up to disinfection. The most sterile is sterilization. And you can just look at what it says, destroys all microorganisms and endospores. So this is a big one. Does it kill endospores? We can't always kill endospores because when we're trying to kill endospores on somebody's skin, you're also killing their skin cells. It's a whole game of what is going to work the best, what's also safe for the patient. So this disinfection kills vegetative cells. Decontamination is also another word for that is sanitation. And it removes organisms to a mostly safe level. So here's some examples, and so you can look through those. And then I just did a little picture here of aseptic versus antiseptic. We use aseptic technique in the lab all the time, right? We use our aseptic technique. So that means without organisms. But can you use aseptic technique on a person? You can't. You have to use antiseptic techniques. So you're trying to get rid of the organisms without harming your patient. Here, we're trying to completely get rid of all the organisms. So those are a couple more terms. And then how we prevent. Remember, we had prevention, identification, and treatment. So our control was prevention, identification, and treatment. So for prevention, we can use chemicals, or we can use physical control, or we can use vaccines for prevention. So what is physical control or mechanical control, and what is chemical control? Let's go through those. Oh, and then here's a couple more terms that I want to talk about is bactericidal versus bacteriostatic. So bactericidal is like suicide. It's like you're killing the bacteria with something like an autoclave. Bacteriostatic is what we do every single day when we put stuff in the refrigerator. So can you kill bacteria in the kitchen? Yes, you can, but you can't cook everything. You know, some stuff you have to use fresh vegetables. You have to use, so to do stuff like that, you have to use bacteriostatic techniques. So that's something to think about when we get into antibiotics. Are these antibiotics actually killing the bacteria, or are they slowing the rate of binary fission to an amount that hopefully your patient's immune system will kick in and your patient will be healthy because their immune system has time to get rid of the infection. Okay, so bactericidal, bacteriostatic are the two that we talk about. Okay, so here's our physical or mechanical control. Physical, mechanical, they both mean the same thing, and these are the ones that we're gonna memorize. So, physical, or sometimes it's called mechanical. So the ones that we're going to memorize are moist heat. And the reason why I wanted you to memorize moist heat is because that's what an autoclave is. So an autoclave, what it does is, you know, we want to try and get rid of endospores. How do we do that? We increase water to boiling. Well, boiling temperature doesn't kill endospores. It has to be above boiling. Well, how do you get above boiling temperature? What would be the physiology of that? How would you do that?
Pressure.
Yes, awesome. So you have to do it with pressure. You have to add pressure to an enclosed environment to increase the temperature, increase moist heat up to a level that will kill endospores. So moist heat, you can also, we can consider that boiling, we can consider that pasteurization, we can consider that autoclave. And I don't want you to memorize any of these things, but I do think it's a good idea for you to just read them yourself. Like, okay, what is boiling temperature? What is autoclave temperature? What is the PSI, the pressure that's needed for an autoclave? How does an autoclave work? You know, that kind of thing I think is important just to know as if you're going into clinical medicine at all. Or, you know, if you're gonna be a dental hygienist have to know that. Okay, so, the next is dry heat, but we're not going to memorize that one. We don't really use that very often if we're not, you know, in actually the clinical setting. They will maybe use that when they're sterilizing instruments on their own.
Is that what we're doing when we put the little?
Yeah, that is what we're doing. We're using dry heat. Yeah, perfect. Yeah.
This is kind of on topic, not off topic, but
Yeah.
do you know the difference between an autoclave and a plasma sterilizer?
I don't know what a plasma sterilizer does.
Okay.
Do you know what it does? Yeah.
This is sterile processing technician. I used to sterilize surgical instruments.
Oh, amazing.
Yeah, it was about 5 years ago, but, so like scopes would go into that type of autoclave. So like there's plasma and there's one that's hydrogen peroxide. So like Da Vinci scopes, laparoscopic scopes, those type of scopes can go into the actual autoclave.
Do you know what it does? What the physiology behind it is?
I mean, it's
Okay, yeah, I'm gonna Google that, cause that is interesting to know, yeah, I don't know the difference.
She scopes and laparoscopic and anything that we couldn't put in moist.
Right, that makes sense, okay, awesome,
But it's pretty interesting.
yeah,
Still have my book. I still
super
have my
interesting. Cool, yeah, if you have anything to let us know, that'd be great.
Yeah.
Okay, so cold is another physical mechanical control. Desiccation, so desiccation means that we dry it out. So like think of like beef jerky or something like that, is you dry something out to prevent bacterial infection, so that the bacteria can't reproduce in a dry environment. Radiation, so obviously we don't really use radiation ourselves, but they use radiation in sterilizing some instruments in a clinical setting. UV radiation, so this is a whole new thing, we're actually gonna do this ourselves in the lab. We're going to use UV radiation to prevent the binary fission of bacteria. For now, just know UV radiation is a method of control, but I'll explain to you how it works when we get to that lab. Yeah.
Is that like the stuff you can buy where you can put your phone in the little light
Yes.
case or whatever?
Yes, and they're using that a lot in like a clinical setting. They'll just turn a UV light on, leave the room, and it's using UV light. But we'll talk about the effectiveness and all that kind of stuff when we do that lab. Yeah.
Are we also gonna get into the post-surgery surgical respiratory droplets in the lab?
No.
I was just curious why that is considered working through the air despite droplets being a liquid, or if that was just a convention that because the medium is air that the droplets transfer through.
So how UV radiation is effective, you mean at that? So.
I'm misunderstanding a fundamental. Sorry.
Oh, no, that's okay. No, yeah. So UV radiation is used to control airborne, right? Things that are airborne.
Right.
Because how else would you spray in the air? Like what are you gonna do to wipe it down? What are you gonna do? It's airborne, so that's a good methodology to use. Filtration also, there is a lot of new technology with filtration, so they're using those a lot of times now. And then osmotic pressure, and the reason I wanted you to memorize osmotic pressure is just because that's what we do at home. We add a bunch of sugar as a preservative or add a bunch of salt as a preservative, and preservative, what you're preserving is the original product. And you're preventing the bacteria from eating it and making it rancid, just like Louis Pasteur did. So what you're doing is you're creating an environment where if you have tons of salt or sugar out here, then remember water follows salt. The water from the bacteria will leave and desiccate or dry out the bacteria, so the bacteria will die or will be most likely in a stasis position until some of the bacteria that survive can eat around it and form acids. I don't get too crazy. Yeah.
So you kind of think of bacteria as liking sugar,
Yes.
but are you talking about?
Good point. Yes, no, they do like sugar, but just because there's such a high concentration, it makes the water leave their cells.
So are we talking about like strawberry jam where it's almost
Yes.
all sugar?
Yes.
Okay.
Yeah, and really, if you had a soda left out on the counter, how long would it last? Months? There's so much sugar that the bacteria that live in there, it will dry them out right away because the water will leave their cytoplasm, and then they won't survive in that environment because it's so thick. So that's the same thing that people do with salt, is if it's a high salty food, they're preserving so that the bacteria can't grow in that environment. Okay, so that's osmotic pressure. Okay, now here we're moving on to chemical control, and we're not gonna memorize all these either. Here's the formula. Sterilization, because that's the most stringent, and you can't use that on patient skin. So it wouldn't be a sterilization technique. Alcohol is irritating to the skin but necessary to prevent infection. Maybe. C, the patient's skin must be scrubbed for an adequate amount of time to allow the alcohol to kill microbes that are present. Maybe. D, alcohol inhibits production of microbes present on the skin, though it cannot kill what is already present. That one doesn't even make sense, right? How can it prevent new infection but not kill what's already there? So A and D don't make sense. B and C are two babies. So alcohol is irritating to the skin but necessary to prevent infection is what I would have put. But what the answer is C. It must be scrubbed for an adequate amount of time to allow the alcohol to kill the microbes that are present, and the reason is because alcohol evaporates, it's volatile. So it will evaporate, and so if you wipe it once, is it at its full effectiveness? So what I would say is, yes, it is irritating, but the most accurate is C. So you just have to get used to answering these questions, and you have to do process of elimination. Yeah.
Your slide before said that alcohol is non-irritant.
So alcohol is a non-irritant, which, but that's the thing that would trip me up, is because if
I
you're,
don't think it's gonna irritate the cells itself like hydrogen peroxide does, but it does sting to an open.
yes, it does sting to an open wound, but not to closed, to a, there's a word for it, like intact.
Cells.
Yeah, yes. So really, it should be C, but in my mind, that would have been a tricky question to answer. So just get used to answering these tricky questions, because generally in the NCLEX or examinations at all, they'll have two that are both right or semi-right. Which one is the most right? So just practice answering those questions. There's tools on your phone. If you go through these, maybe just take like one day during Thanksgiving break and write, type out all the questions, or take screenshots of all the questions and write down what the correct answers are, so that you can start practicing. Because all of the free practicing is the best. The more you practice, the better. Okay, so our bacterial vaccines. So we're gonna talk a ton about vaccines when we talk about the immune system, and what are vaccines doing, and why they are effective versus non-effective, what a vaccine is, what it does, how it works. So we're gonna go through the whole thing of vaccinations when we get to viruses, but I'm just gonna introduce it a little bit here. So I wanted to bring up a PubMed paper. So this is what a journal article looks like. If any of you are interested in research, let me know. This is what it looks like when you Google something, when you PubMed search. This is what will come up. And so this is an article written by these researchers that says vaccination against bacterial infections challenges progress and new approaches with a focus on intracellular bacteria. So the intracellular bacteria, what was one of the intracellular bacteria that we talked about? Do you guys remember from your note cards? I don't blame you if you don't, because we haven't studied them yet, really. Do you guys remember the one that's intracellular always? Chlamydia? Yeah. Yes, awesome. Yes, Dr. Winters had it. So chlamydia. So some bacteria that live inside cells, how do we get to them if they're inside of a given cell? I just thought it was kind of a cool thing to bring up, but it's a good idea anytime that you're having questions to PubMed ask. Okay, so here are the bacterial vaccines, and I'm going to put that on our Thursday slides to explain to you what all of these are. Bacterial vaccines, because I think this will help you start memorizing. When you start memorizing your bacteria, all of your bacterial diseases is the ones that you've been vaccinated for, and why we vaccinate for those ones. So this is diphtheria, tetanus, acellular pertussis, Haemophilus influenza, pneumococcal conjugate from, I'll tell you about what it is later. And then here's meningococcal. And we'll talk more about why it's important and why they're crazy. I did already put a slide on there, that's convenient. Okay, So, for our bacterial vaccines, we have our pneumococcal, meningococcal, typhoid. So typhoid, you can get a typhoid fever vaccine, but most people don't get that because we're not in an area where typhoid fever is common. One thing that I just want to throw out there, that Salmonella typhi is not Salmonella typhimurium. Those are two different. Salmonella typhimurium is from chicken. Salmonella typhi is typhoid fever. Two totally separate things. So they just have the same genus, but they're two, and they have almost the same species name, but they're two very different bacteria. So Salmonella typhimurium is the one you'll come across over and over and over. That's the one that people get from eating chicken that's undercooked. Okay, so pneumococcal is strep pneumo. So strep pneumo is Streptococcus pneumoniae so do you guys remember what the name of the bacteria is called that's a group A strep? Does anybody remember? You had group, it's,
No, Streptococcus.
yep, Streptococcus. That's okay, it's a hard one. Yeah, we haven't really talked about that yet very much. I mean, we just did it, Pyogenes, yep. So Pyogenes, I don't want to go into that. So, that is strep throat. That's from our note card. That's a group A strep. And then, I should write out strep, because now you're gonna need staph.
And that's the only group A strep, right?
Yes.
Sorry, what is group A mean?
Anything else is a group B strep. So group A, they just categorized them. If it's group A, it's this. If it's group B, it's every other kind of strep. So every other kind of strep, we classify a group A strep as strep pyogenes because it's in its own world and it's super virulent. A lot of people get strep throat. It's all over the place. It's unique than the other Streptococcus genus, all the species that are in the other Streptococci. So everything else is a group B strep. But strep pneumoniae is a group B strep, and that is the pneumococcal. But the meningococcal is because strep pneumoniae, and what's our meningococcal is meningitis. Neisseria meningitidis. Those can cause meningitis. So meningitis, if it's bacterial, is deadly. If it's viral, it's not deadly. Most cases of meningitis are viral, but we treat everyone with antibiotics that has meningitis just in case it's bacterial, because it has a huge mortality rate associated with meningitis. So these two bacterial organisms are the ones that are most likely to cause meningitis, so we vaccinate against those. So we have those vaccinations. You know, it's hard to get the vaccination just right, and I'll teach you about vaccinations and what they are. It's hard to get them just right, and so we don't have vaccinations for everything, obviously. So those two we do. We put billions and billions of dollars into getting them just right because they're extremely deadly. Okay, so then DTaP or Tdap are the vaccines